Welcome, learners. I am Dr. Seema Desai Fernandez, Assistant Professor, Department of Botany, St. Xavier's College, Mapsa, Goa. This program is for TY Botany student, Semester 5, Paper Code BOC 106. The title of the unit, Unit 3 Cell Division, Module name Overview of Cell Cycle, Module number 26. Outline Overview of cell cycle. In this, you are going to learn types of interphases and M phase and cell cycle diagram. Learning outcomes describes the types of interphases and M phase of cell cycle, explains the cell cycle diagram. Introduction. Definition of cell cycle. The cell cycle is the process by which a cell replicates its genetic material and synthesizes the other elements of the cell. And at the end, it is divided into two daughter cells, which is known as the cell cycle. Fundamentally, the cell is divided into two basic phases, interphase and mitotic phase, M phase. Interphase is the phase in which cell grows and makes its copy of the DNA. Interphase is basically called the resting phase of the cell because during this period the cell prepares itself for cell division by experiencing both cell growth as well as DNA replication. The interphase is further divided into three phases. G1 phase, known as the gap 1 phase, S phase, that is DNA synthesis phase, and G2 phase is the gap 2 phase. This figure explains the three phases of interphase, that is G1 phase interphase, which shows the checkpoint, that is also called as the restriction point. S phase, which is also called as the DNA synthesis phase, and G2 phase, which also shows the checkpoint. The interphase, which is showing all three phases, G1, S phase, and G2 phase, after the G2 phase prepares the cell to go into the next phase called as the mitotic phase. Mitotic phase or M phase is the division phase in which parent cell is divided into two daughter cells. Here, the cell separates its DNA into two sets and divides its cytoplasm, forming two new cells. There are four distinct phases: interphase, G1S and G2 phases, which are known as the interphase phases. The prefix inter means between, reflecting that interphase takes place between one mitotic M phase and the next. Mitosis M phase is a nuclear division phase. Hence, it is called mitosis, followed by a cell division called as cytokinesis. Let us go into the G1 phase or G phase. It is also called as the first gap phase. In this phase, there are metabolic changes prepare the cell for division. The cell grows physically, larger, copies organelles, and makes the molecular building blocks it will need in the later steps. At a certain point, called as the restriction point, the cell is committed to division and moves into the next phase called as S phase. S phase is also called as the synthesis phase because here the replication of DNA will take place. In S phase, the cell synthesizes a complete copy of DNA in its nucleus. Each chromosome now consists of two sister chromatids. DNA is manufactured during S phase. It also duplicates a microtubule organizing structure called the centrosome. The centrosome help separate DNA during M phase. Next phase is the G2 phase, that is, it is also called as the second gap phase. Here, the cell prepares itself for mitosis. During G2 phase, the cell grows more, makes protein and organelles, and begins to recognize its contents in preparation for mitosis. G2 is usually shorter than G1 and is usually 4 to 6 hours. G2 phase ends when mitosis begins. This is the figure which explains four distinct phases. Let us see a little bit in detail the G1 phase. G1 phase, in this phase, the cell metabolically gets activated. It duplicates organelles and cytosolic components, starts replicating the centrioles, starts replicating centromeres. The S phase shows the DNA replication, and here each chromosome, that is here the S phase shows DNA synthesis, and each chromosome now consists of two sister chromatids. The G2 phase shows cell growth continues in this phase, enzymes and other protein are synthesized, and replication of centromeres is completed. M phase, a nuclear division phase, starts. The cell prepares itself in G2 phase and then it goes into the next phase, that is the mitotic division phase, followed by a cell division called as cytokinesis, and it forms two daughter cells. Mitosis precedes the cytokinesis. Now here in the diagram there are checkpoints everywhere, checkpoints in G1, then checkpoint in G2, and there is a checkpoint in mitosis. These checkpoints checks whether the cell has completed its process and now the cell is ready to go into the next phase. Let us see the mitotic phase. M phase is the mitotic phase in which multi-steps perform to duplicate chromosomes, separate and move into the new similar daughter cells. Mitosis takes place in four stages: prophase, sometimes divided into early prophase and prometaphase. Then it goes into the metaphase, anaphase, and the telophase. Mitotic phase or M phase, in this, it shows the nuclear DNA of the cell condenses into visible chromosomes and it is pulled apart by the mitotic spindle, a specialized structure made out of microtubules. There are two parts. The first part is called as the karyokinesis. It is also called karyokinesis, means nuclear division. And the second part is known as cytokinesis, means the cytoplasm division, in which the cell splits into two daughter cells. Cytokinesis usually begins just as the mitosis is ending, with a little overlap. Importantly, cytokinesis takes place differently in animals and plant cells. The cell cycle checkpoints. The cell cycle checkpoints determine if a cell is ready to progress to the next stage. Late in the G1 phase, the G1 checkpoint determines if the cell will enter the following S phase. G2 checkpoint determines if the cell will enter the M phase and requires the proper completion of DNA synthesis. The third cell cycle checkpoint is the spindle assembly checkpoint, which occurs between metaphase and anaphase and requires the proper attachment of all the chromosomes to the spindle apparatus. Cell cycle exit and G0 phase. Some types of cells divide rapidly. Some types of cells divide rapidly, and the daughter cells immediately undergo another round of cell division. For instance, many cell types in an early embryo divide rapidly, and so do cells in a tumor. Other types of cells divide slowly or not at all. These cells may exit the G1 phase and enter a specialized resting state known as G0. In G0, a cycle is not actively preparing to divide. G0 is okay. In G0, a cell is not actively preparing to divide. G0 is a permanent state for some cells, while others may restart division if they get the right signals. Different cells take different lengths of time to complete the cell cycle. Function of the cell cycle. The most basic function of the cell cycle is to duplicate accurately the vast amount of DNA in the chromosomes and then segregate the copies precisely into two genetically identical daughter cells. This process defines the two major phases of the cell cycle, that is interphase and the mitosis. These are the references for this module. Thank you, students.